Hello everyone, it's an honor to present our study. Characterization of Mycobacterial Kinase PknG substrates reveals the PknG-KARS-MAPK phosphorylation signaling axis in the host. Mycobacterium tuberculosis causes tuberculosis. Its virulence factors help the bacterium survive inside host cells and evade immune attack, leading to persistent infection. However, how these factors interact with the host is not fully understood. Our study has 4 highlights. First, we used a multi-omics strategy, proteomics, phosphoproteomics, and interactomics, to study PknG substrate regulation in host cells. Second, in vitro phosphoproteomics identified that PknG directly phosphorylates KARS at T592 and T595. Third, PknG disrupts the downstream MAPK/NF-kappa B pathway via the KARS phosphorylation axis. Fourth, in mice, PknG overexpression caused lung injury, inflammation, and T-cell reduction leading to immunosuppression. To investigate PknG's effects, we transfected PknG into HEK293 T cells and performed TMT quantitative proteomics and phosphoproteomics. The regulated phosphoproteins were highly enriched in RNA-related functions. We then performed interactomics on the same samples. The interacting proteins were mainly involved in ribosome RNA transport and aminoacyl-tRNA ligase activity. Integrating the omics datasets, we identified 16 potential substrates. Among them, only lysyl-tRNA synthetase, KARS, was strongly linked to both ribosome and RNA transport. To confirm PknG-mediated phosphorylation of KARS, we carried out in vitro kinase assays with recombinant proteins followed by high-resolution mass spectrometry. PknG strongly phosphorylated the C-terminus of KARS. Since the C-terminus contains multiple serine/threonine residues, we performed site-directed mutagenesis and found that T592 and T595 are the critical phosphorylation sites. This was validated using PknG from Mycobacterium tuberculosis. Next, we asked how T592/T595 phosphorylation affects KARS function and downstream signaling. In vitro activity assays showed that phosphorylation at these sites enhances KARS enzymatic activity and promotes TNF-alpha receptor release, thereby modulating the MAPK phosphorylation pathway. Thus, PknG acts in a KARS-dependent manner in the host. To model the virulence factor's physiological role, we infected mice with PknG overexpressing strains. H&E staining and qPCR revealed severe lung injury and inflammatory changes. Finally, to assess immune function, we analyzed spleens from infected mice by flow cytometry for antigen-specific cytotoxic T cells. PknG overexpression induced a strong immunosuppressive effect. Our study provides a comprehensive proteomic and phosphoproteomic landscape of PknG in host cells. We discovered that KARS not only serves as an adapter for protein synthesis but is also a key phosphorylation substrate of PknG. Dynamic phosphorylation at T592/T595 regulates the MAPK pathway, impacting lung pathology and immune function in mice. This reveals a novel mechanism of immune evasion by Mycobacterium tuberculosis.